records to show that. In Egypt, Babylon, Rome, and China, people produced wine from grapes and beer from malted barley. In South America, people produced chicha from grains of fruits, mainly maize, while in North America, people made octli from agave, a type of cactus. At the time, people knew that leaving fruits and grains in covered containers for a long time produced wine and beer, but no one fully understood why the recipe worked. The process was named fermentation, from the Latin word fervere, which means to boil. The name came from the observation that mixtures of crushed grapes kept in large vessels produced bubbles, as though they were boiling. Producing fermented beverages was tricky, however. If the mixture did not stand long enough, the product contained no alcohol. But if left for too long, the mixture rotted and was undrinkable. So this was before Christ. Now, fast forward to 1856. A man named Bigo sought Pasteur's help because he was having problems at his distillery, which produced alcohol from sugar beetroot fermentation. The contents of his fermentation container were embittered, and instead of alcohol, he was obtaining a substance similar to sour milk. Pasteur analyzed the chemical contents of the sour substance and found that it contained a substantial amount of lactic acid instead of alcohol. When he compared the sediments from different containers under the microscope, he noticed that large amounts of yeast were visible in samples from the containers in which alcoholic fermentation had occurred. In contrast, in the polluted containers, the ones containing lactic acid, he observed much smaller cells than the yeast. So Pasteur figured out that it is indeed fermentation, and it is of two types, alcoholic and lactic acid. So Louis Pasteur, a French chemist, was the first to demonstrate experimentally that fermented beverages result from the action of living yeast transforming glucose into ethanol. Moreover, Pasteur demonstrated that only microorganisms are capable of converting sugars into alcohol from grape juice, and that the process occurs in the absence of oxygen. He defined it as respiration without air. Pasteur went ahead and published his results in 1857, which was titled, I really don't know French, and I'll try. I've been to France though, Mémoires sur la fermentation alcoolique. So, even though Pasteur thought there were two types of fermentation back in 1800s, now we know there are five: alcoholic, lactic acid, propionic, butyric, and mixed acid. My hope is to go over them a little by little. I will probably delve more into alcoholic fermentation and cover a little bit of all of them, just because I know it's part of your curriculum. Why alcoholic fermentation? As you guys already know, I am in beverage industry. We don't do alcoholic beverages yet, but fermentation is something we study very thoroughly. So moving on, alcoholic fermentation generally means production of ethanol. You all know the formula. Commonly, yeasts, particularly Saccharomyces cerevisiae, are used for production of various alcoholic beverages as well as industrial alcohol. Yeast are essential aerobic organisms, but you all know that they can also grow as facultative anaerobes. This is a simple chart to show how alcoholic fermentation takes place. So let's delve into this a little bit. Conversion of pyruvic acid to ethanol proceeds in two steps. If you see here, pyruvic acid to acetaldehyde and acetaldehyde to ethanol. The first step is catalyzed by pyruvic acid decarboxylase, which requires TPP, which is thiamine pyrophosphate, as coenzyme, and the second step by alcohol dehydrogenase, which requires nicotinamide adenine dinucleotide 2 as coenzyme. I'm sure you all are familiar with coenzyme. Let me say two sentences about it. So what is a coenzyme? It is a non-protein compound that is necessary for the functioning of an enzyme. It actually binds with the enzyme, and that's how an enzyme works. So in this case, for alcoholic fermentation to happen, it goes through two steps, and two coenzymes help to make alcohol. Um, Various strains of yeast, mostly belonging to Saccharomyces cerevisiae, have been developed and carefully selected for large-scale manufacture of alcohol for different purposes. Also, various materials and conditions are used depending on the nature of the product desired. Let me give you an example. For production of baker's yeast used in bread industry, I don't know how many of you bake. I bake very regularly. In fact, in my bread machine right now, rosemary bread is being baked. I wish you could smell it. So anyway, for production of baker's yeast in bread industry, strongly aerated cultures favor large cell yield with little or no alcohol. Extract of malted barley serves as substrate for beer production. So as I said, I will probably speak a little bit more about alcoholic fermentation. First of all, it's the oldest one known to mankind. Second, it is for all practical purposes the easiest of all. So the starting material contains large amount of maltose, which is a disaccharide of 2 glucose units. So what happens is, so we take barley seeds, there's starch in seeds, the starch undergoes hydrolysis, that hydrolysis produces maltose. So got it? So now when we have maltose, that gets split into glucose, that becomes a substrate for alcohol fermentation. So for example, let's take wine. In wine, grape juice is the substrate of choice. Specific selected strains are employed to impart characteristic flavor and taste of different alcoholic beverages. For manufacture of industrial alcohol, we all know how industrial alcohol has been in the news recently with COVID. Everyone wants to make a sanitizer. Generally, molasses is used as the starting material for the industrial alcohol. Also, sulfite liquor, which is a waste product of paper industry, is used as a cheap substrate for industrial alcohol production. An obvious question here is, since we're not doing this interactive, we talked about fermentation, alcoholic fermentation yeast. Can bacteria do alcoholic fermentation? Definitely. From, science progresses, now we know that some bacteria, especially Zymomonas mobilis and Pseudomonas sacrofolia, are also responsible for conversion to ethanol, pyruvate conversion to ethanol. So let's move on to fermentation number 2, which is lactic acid. Before we delve into what lactic acid fermentation looks like, let's try to understand a little bit on lactic acid bacteria. I'm sure you all eat yogurt, dahi, and we all are aware of lactic acid. But let me tell you this, being in the food industry, and I don't do dairy, we don't do dairy, my company. So lactic acid bacteria is a pain. In the manufacturing operations, you don't want lactic acid bacteria. So we do study this quite a bit just because you don't want it. So lactic acid bacteria, now we know, are both morphologically and physiologically very diverse. We'll learn later in these slides that lactic acid fermentation is of 2 types, homo and hetero. So homo-fermentative lactic acid bacteria are quite a few, lactis, diacetylactis, thermophilus, bulgaricus, acidophilus. Hetero-fermentative is a very, something that is rampant in industrial production is Leuconostoc mesenteroides, Lactobacillus brevis, and bifidum. There is also a spore-forming lactin bacteria. The name is right there on the slide. As I said, lactic acid fermentation is of 2 types. One is homo-fermentative and second is hetero-fermentative. Lactic acid in homo is produced as the sole product by reduction of pyruvic acid. In hetero, the products are lactic acid and ethanol, or acetic acid and carbon dioxide. In easier terms, that's all you have to remember about lactic acid fermentation. 2 types. In one, the sole product is lactic. In second one, the products are lactic acid and ethanol, depending on the route it takes, or acetic acid and carbon dioxide. So homo-fermentive, right in front of you, easy peasy, from pyruvic acid to lactic acid, using the enzyme lactate dehydrogenase. The reaction also regenerates NAD from NADH2, the coenzymes as I was talking about. And when it comes to hetero-fermentative pathway, it is more complicated than Homo. And it does produce volatile acids, carbon dioxide, and lactic acid. And you see it's right in front of you, all the different coenzymes that are operating here. So we move on to propionic acid fermentation. I don't know how many of you are aware of propionic acid, but again, coming from the context of food industry, propionic acid is one of the first substance that was granted GRAS status. So GRAS is known as generally recognized as safe. FDA grants that status to certain ingredients, which means these ingredients could be used in food industry or pharma or beverage industry without any potential side effects. So it's a big deal getting a GRAS status for an ingredient. Propionic acid is also a very well-known preservative. You know, it's a wonderful ingredient that the food, pharma, CPG space use quite a bit. So propionic acid is produced by several anaerobic bacteria, among which are Clostridium, Selenomonas, etc. They are natural inhabitants of rumen of herbivorous cattle. Something interesting I found on this one. I'm sure most of you like cheese. I'm not a big fan of Swiss cheese myself because it has this typical, very aged flavor. But where that is coming from is actually propionic acid. So lactic acid bacteria and propionic acid bacteria, they make Swiss cheese, and that propionic acid is what is used. It contributes to that special flavor. And again, I don't know how many of you have had fondue. That special flavor in fondue comes from this acid. Just a trivia fun fact for you all. So I tried to find something fun. This diagram was very complicated. So I tried to find something fun for you guys today. The propionic acid bacteria, what it does, it disseminates glucose. You know how you've seen every process fermentation starts from glucose. So it produces pyruvic acid. There is a carboxylation reaction. Pyruvic is converted to oxaloacetic. It is then reduced to succinic. After it gets to succinic acid, it is then converted to succinyl coenzyme. From there, it gets to propionyl coenzyme through very enzyme-linked catalysis. It is this is where it gets pretty complicated. But however, in the final step, propionyl CoA yields propionic acid, and that's how we get our acid that I said is very useful in food industry. So also in the final step, coenzyme A is transferred to succinic acid by an enzyme, which is coenzyme A transferase. This is a very simple diagram, but it kind of shows you how molecules are moving. If you see when glucose starts at 3 molecule, lactate at 3 molecule, and it converts to 4 molecules of propionate. Moving on. So next one is butyric acid or butanol fermentation. The bacteria carrying out this fermentation are all obligately anaerobic spore-forming bacteria belonging to the genus Clostridium. Besides butyric acid and N-butanol, several other products of this fermentation are acetic acid, ethanol, isopropanol, and acetone, which obviously depends on the species that is being used. For example, some of the bacteria like Clostridium lactoacetophyllum or Pasteur will produce butyric acid with acetic acid. Some of them will combine to produce acetone. No matter what, as a fermentation product, carbon dioxide is always present. Again, this is a roadmap showing how butyric acid is formed. You see, it is a complicated process, and it starts with glucose, but it has a lot of, it's like the process of distillation. So you get ethanol, acetic acid, acetone, isopropanol, butyric acid, and butanol. It goes through a series of a series of enzymatic transformations, a series of, you see how hydrogen is coming in and acting with the coenzyme A. So all of that happens. But again, something to remember, Clostridia always produce molecular hydrogen as one of the fermentation products when it comes to butyric acid fermentation. All right, we are almost at the end of this all. The last one is mixed acid fermentation. This type of fermentation occurs characteristically in bacteria belonging to the family Enterobacteriaceae. These bacteria can grow both aerobically, carrying out respiration, or anaerobically, carrying out fermentation. This type of fermentation is called mixed acid because several different organic acids and neutral compounds are produced. A characteristic acid, although, is formic acid, even though it is really not the major product in this one. Depending on species, different substances are formed like acetic acid, succinic acid, lactic acid, ethanol, acetone, butanediol, carbon dioxide, molecular hydrogen. So the list goes on and on. On the basis of fermentation products, the enterobacteria can be divided into two groups. One group having an E. coli type fermentation and the other having an Enterobacter aerogenous type. There is a significant difference in these two types. When Enterobacter is working, we get acetone and butanediol, but in case of E. coli, in case of E. coli, these are absent. But at the same time, just like every other fermentation, it also starts with dissimulating glucose to pyruvic acid, as you just see here. See how complicated this is. It starts with glucose, converts to pyruvic acid, takes a different, it's like different branches of the tree, but it is producing lactic acid, formic acid, acetic, ethanol, butanediol, acetone, formic acid, succinic acid. One thing to remember in this one, compared to the last one, this is the only fermentation that is producing formic acid. Again, the yield is not very high, but this is the one that produces formic acid. So I guess that's about it. But as I was researching into this topic, I did find a quote. I also found out that there is a fermentation association. So, if you have time, you might want to check it out. And I found this very interesting quote from David Rains Wallace, which says, fermentation may have been a greater discovery than fire. And you know why? Because in the past, again, those many centuries back, when there was no refrigeration and not any wonderful means of preservation, fermentation preserved the food. Fermentation provides subsistence. So very important process. Produces nutritious products, produces wonderful ingredients for pharma company, for food company. So I hope you get to research into fermentation a little bit. I do thank you for your time, and you will see in some of my slides, I have a lot of references here. In my team, I have a brilliant PhD in microbiology, and she actually helped me put this together. And she gave me a few references of the books as well. You can buy them on Amazon. That's about it from me. Thank you so much for your time.
Thank you so much, Dr. Jagrati. Any questions, audience? If anybody want to ask any question, please unmute yourself and then ask your question.
And just to add, if you can't think of any question right now, and if you need any help, feel free to get my email address from Shalini, and I'll be more than happy to respond.
As we are waiting for any query to come, Jagrati, can you enlighten these young students? How can they prepare for their career since they are going to pass out from their college? So how they can enhance in their degrees and everything? You are muted. Please unmute yourself, Jagrati.
Oh, all right. Thank you. So I guess the biggest thing to remember here is what you want from your life and what are you passionate about. I'll tell you this thing. There's no substitute. You must have heard it over and over again. My dad told me that all my life. He still does. There is no substitute to hard work. So yes, you can work smart. You don't have to work hard if you work smart, but there is just no substitute to hard work. They say garbage in, garbage out. So what you put in towards your career is what comes out of it. I think that answers that question.
So no question is coming out. Yes, ma'am. Ashant, should we conclude now?
Okay.
Yes,
Hello, ma'am. Yes, Dr. Manisha. I would like to ask a question to the guest speaker. Can I ask a question now?
Yes, Manisha, ma'am, please go ahead.
Okay. Hello, ma'am. I would just like to ask you, you being the head vice president of our organization, so I'd just like to know what are the different projects or what work in research are you doing right now? So this will be us to.
So, hi, thank you for the question. There are some network issues, so I couldn't hear all of it, but I am assuming you asked
Audible?
me what are the different research projects
Yes.
I'm working on, right?
Yes.
So my team has different components. One of the team that I lead is called Technical Advancement Team. We are responsible for not just so our strategic projects are 3 to 4 years out, but that team is responsible to guide the strategy 10 years out. So we're working on projects that will come to fruition in 2030. That is my favorite part of what I do. And within part of that project, we are looking into everything. We are looking into green packaging, which is recyclable packaging, sustainable packaging. We are looking into natural ingredients like natural preservatives, natural colors. As I said, we are now delving into fermentation technology. How do we filter it so we get better product? A lot of specifics, unfortunately, I can't provide because of the proprietary nature of the research. But we do have a full spectrum of analytical lab, a physical characterization lab. We are working on getting a micro lab up and running. But really, I mean, what I do like about working here is the flexibility, and it is extremely research oriented. The team is very bright, and they are extremely research oriented. Does that answer your question?
Yes, thank you so much, ma'am. Thank you so much.
You're welcome.
Ma'am, can I ask a question, please?
Sure.
Yes, please go ahead.
Ma'am, first off, good morning to all. And my question is that, is any difference between the anaerobic respiration and fermentation? Am I audible?
Hello.
Am I audible?
Yes, Zubair.
Yes.
No, you are.
Good morning,
Can
ma'am.
you-
Good morning,
Yep.
all. Ma'am, my question is that, is any difference between anaerobic respiration and fermentation?
Yeah. I mean, thank you for the question. So, I mean, I can see where your question is coming from because fermentation does happens without oxygen, but there is difference between anaerobic respiration and fermentation, which is, as we looked into those roadmaps for the processes. So fermentation, as we saw, ends with glycolysis. But anaerobic respiration, it creates pyruvate, and then it continues onto the same path as aerobic respiration. Does that answer your question?
Thank you.
Any more questions? Anyone?
It is a wonderful presentation. Myself want to ask are you in making some kind of product also, or a simple extraction of these kind of alcohols and certain kind of fermentation processes? Means certain product is also there. When you
Yeah, I guess I didn't make it very clear what I work on. So I work for a beverage company and we do make products. Our products are flagship brand is Sparkling Ice. So it is a carbonated fruit flavored beverage. I think last time I was in India, I saw something similar. Is it clear? Is that the brand which is carbonated beverage fruit flavored? So that's what we do. We are in 25 countries in the world. But we also do other beverages which have immunity benefits or vitamins in them, antioxidants in them. We don't really do ingredients, so we don't do fermentation on site. We learn fermentation because that helps us in developing products. But we are a very retail oriented a brand in the US.
Anyone questions? So I think there are no more questions. Archana?
Ma'am, one more question I want to ask you. I'm Zubair.
Yes, Zubair. Yes, Zubair. Go ahead.
Ma'am, can you please tell us about the applications of fermentation?
So, Zubair, if you saw my first slide, which was kind of a guess what's the topic slide, is, what does fermentation do? There are quite a few applications. I think what comes to my mind, and it comes to my mind because, in my master's program, as you know, I did my master's in environmental sciences. And in one of the labs, we utilized fermentation for treatment of wastewater. So we had, I hope you all know Pantnagar University, and there's a paper mill. And we used to get samples from that paper mill. And, we had a nice big micro lab. And some of my colleagues did utilized fermentation to treat the wastewater. And then obviously, as I said, that slide should tell you it's used in making beverages, especially alcoholic beverages, bread making, kefir, yogurt, pharma company, your penicillin.
Hello. Hope any more query from the participant sides?
Dr. Sharma, please switch on your camera now for a few moments.
Is this good?
Yeah.
Yes.
Dr. Shalini, I would like to invite you for the vote of thanks, and thank you, Jagrati, for giving us most precious time and enlighten my participants by your lecture. Thank you so much. Dr.
Anytime.
Shalini?
It is my privilege to propose a vote of thanks on this occasion. I, on behalf of Department of Biotechnology, extend a hearty vote of thanks to Dr. Jagrati Sharma who blessed us with her presence and took out her valuable time out of her busy schedule on a very short notice. I especially like to appreciate her efforts in such busy schedule that she explained this fermentation so precisely. Her way of explanation was exemplary. I hope all the participants were enlightened. And then I will take opportunity to express my gratitude to our principal ma'am, Dr. Shalja Pant, and our chairman, sir, for giving this opportunity to us. And lastly, but not the least, our technical team and Mr. Sujit TK. We extend a big thank to them, to all our fellow colleagues without whom their cooperations, this was not successful. And I heartily congratulate all the participants for successfully attending this event, and hopefully they must have gained some knowledge about the fermentation. Thank you so much, Dr. Sharma, for for gracing this occasion, and thank you so much for all your efforts. Everyone have a nice day, and Dr. Sharma, good night and have a nice weekend.
Thank you so much.
Thank you so much, everyone.